Hi there. So today we are joined by Dr. Jonas Bergquist, who is the director of the Uppsala ME CFS Collaborative Research Center, which is established by Open Medicine Foundation. So today we're just going to talk a little bit about the research progress that is going on at Uppsala University. So great to be joined by you today, Dr. Bergquist.
Thank you, Rebecca. Good to see you, and I'm happy to be in this interview.
Great. So I wanted to start off by talking a little bit about a very fascinating study that you were working on. It's about post-viral complications in herpes simplex encephalopathy, or HSE. And this specific study provides a very unique opportunity to examine a post-viral impact as it progresses compared with ME/CFS disease progression. So I'm wondering if you could just please talk a little bit more about this and why studying HSE patients may be useful for comparison purposes with ME/CFS specifically.
Yes, so actually a few years ago, we got the opportunity to start being involved in collecting patients with other post-viral fatigue syndromes. And one of the patient groups that we could approach was the Herpes Simplexencephalopathy patient group. It's a very, first of all, I want to say it's a very rare disease. Although herpes simplex virus infection is very common, very few patients get problems in the central nervous system of this infection, but some do. And actually we have in our cohort and in our biobank managed to collect the largest cohort in the world as far as we know. So that's an opportunity, of course. And in this patient group, we know that patients, they get an infection, and the infection spreads into the central nervous system. So we also see doing imaging of the brain and also monitoring cerebrospinal fluid, the liquid that surrounds the central nervous system. We can monitor inflammatory reactions in the central nervous system. And this is exactly what we are sort of targeting when we are now monitoring our ME patients, myalgic encephalomyelitis patients. So that's a reason for the interest of the HSE patients specifically. But then we also in HSE have the opportunity and through collaboration to collect the patients from their initial infection and admission to the hospital and the intensive care treatment and monitor them over time. So we have time resolved samples during the initial infection disease, and then over time monitoring the same individuals for up to 2 years. So we have biological fluids collected, and we have all the clinical symptoms and the questionnaires and so on collected over a long time. So this actually makes it possible for us to see what happens in the progress from an infectious disease, a viral disease, and what potentially could lead to, well, neurocognitive issues, more chronic damage to the central nervous system, development of autoimmunity as is one target that we have been monitoring. So absolutely unique opportunity to see something that started early, monitor over time, and then collect information. Now we know these patients been collected for quite a long time, and we now know also the outcome of all the patients. So we know that some of them have totally coped with the situation and now are back into a normal healthy life, while a subgroup is actually ending up with a chronic issue and sequela, so a long-term consequence, and that actually resembles about ME situation. So we hope to learn more from this.
Thanks for explaining that.
And
so,
in the what I wanted to add also in the samples we have collected, which is cerebrospinal fluid, the liquid surrounding the brain, we have blood samples, we have also urine samples. We have done monitoring of inflammatory markers when it comes to proteins. We have monitored the cell damage markers in proteins, but also had the opportunity to monitor small molecules, metabolites. So we have been doing screening of the metabolomic changes in these patients over time. And this is also very much in line with what we do in our ME patient cohorts.
That's fascinating. So I wanted to talk a little bit more about that and the sampling and kind of what you're finding in these samples. Mainly an area of focus that I'd like to discuss more is actually the autoimmune antibodies. So the possible development of these autoimmune antibodies is this consistent finding that you found across HSC patients, long COVID, ME/CFS, and it kind of gives us this window into understanding these symptoms such as neurocognitive disturbances, peripheral symptoms, POTS and pain, other targets of these autoantibodies. So in your study, you actually mentioned how no autoantibodies could be found in the cerebral spinal fluid from 2 ME/CFS cohorts and how this is good news. So I'm just wondering first if you could explain kind of what autoantibodies are, just from the very basics, and why they're important in looking at ME/CFS, and then why it is good news, explaining a little bit more about why this specifically is good for ME/CFS patients, that it's not in the CSF fluid.
Mm. Yeah, so autoimmunity, first of all, is unfortunately a rather common cause of disease, especially in women, unfortunately.
Mm.
And there is a situation that women have generally a more strong immune system. So that's why women also have a higher risk of getting autoimmunity when the immune system start to react against our own cells and structures in the body. So the immune system is supposed to protect us from foreign substances, foreign molecules, foreign organisms, bacteria, viruses, etc. But if you have a too strong reaction of your immune system, you can also cause autoimmunity, that is reacting against ourselves or self-specific proteins and molecules in the body. So the reason why we get these autoantibodies in certain cases is it's been discussed quite a lot. It can be that there are homologies between, for instance, virus proteins and some of our own proteins, and then the immune system start to let's say misdiagnose the molecules in a way. So it actually start to react against molecules that should be our own cells or cell receptors and surface structures on cells, and mistake those structures for being a foreign agent from a virus or bacteria. So that's one potential reason, at least. What we see in patients that are affected by neurocognitive issues, and specifically in, for instance, HSE, the HSE patient group that we talked about, we see autoantibodies against, for instance, NMDA receptors. That's a transmitter receptor that is very important for us when it comes to neurocognition. Those antibodies have been detected in some other neurological diseases also, and we are right now trying to monitor them in ME patients, in post-viral infecting, like post-COVID or long COVID patient groups. In our ME patient group, we, together with other colleagues in, for instance, Germany, Carmen Scheibenbochen's laboratory, have found that ME patients specifically expresses autoantibodies against muscarinic and beta-adrenergic receptors. And those are receptors that are very important for cell-cell signaling and cell transmission and neurotransmission. So the beta-adrenergic receptors react on the adrenergic neurotransmitters, and the muscarinic receptors react on acetylcholine signaling, for instance. And they are very, very important for the most fundamental control of functions in the body. For instance, heart rate and things that are very important for us. So interesting enough, our ME patient cohorts, they have an upregulated production of these autoimmune antibodies. So we see them in circulation. When we take a blood sample, we can detect them with our assay. However, when we look into the central nervous system by analyzing cerebrospinal fluid, we don't see any production of these antibodies. And so we say, okay, there is no intrathecal production, we call it.
Right.
So that's produced inside the central nervous system. There is no evidence of that in our ME group. So why are we happy for that? Well, you could think as a scientist, it would be fantastic to find a marker
Right.
when you look for it in a specific fluid or so. But in this case, we are actually happy that we did not find them there, because if one potential solution of reducing at least the problem for ME patients would be to eliminate those antibodies from circulation or from the tissues by, for instance, plasmapheresis or immune adsorption. And those experiments have been done in a small cohort of patients, and actually with some success at least. If we have them in circulation in the periphery, they are easily accessed, so we can get to them easily, and we can reduce the influence of them by eliminating them by plasmapheresis or immune absorption. If they are in the central nervous system, it's not so easy to get to them, because how should we wash the brain from these antibodies? It's not going to be very easy to do that, unfortunately. But luckily, in this case, we don't see them in central nervous system. And I want also to draw a parallel to multiple sclerosis patients, MS patients, which actually resembles to some extent to our ME CFS patients when it comes to symptoms and also the autoimmunity. In multiple sclerosis, we know there is an intrathecal production of antibodies. So we have autoantibodies produced that are affecting and attacking tissue, and specifically myelin-based protein, for instance, in the central nervous system. So in that case, the treatment has to be different from what we would expect from ME patients when it comes to plasmapheresis or immune absorption, for instance.
Right. Oh, okay. So that makes sense. So in a way, when reading that, we say, well, why is it good news? As you mentioned, because we want to find a marker of some sort. We want to find, we want to be able to nail something down and say, well, this is, something that's specific for ME/CFS. But it sounds like it is good news in the way of treatment targets then, that it would be easier to get to because it is not found in the CSF. So thanks for explaining that a little bit, because I know that was something that it was an interesting finding.
Yeah.
So a lot of your work also has to do with neurointensive care patients, these very critically ill patients. And another initiative that you've taken on at Uppsala is starting a collaboration with the neurointensive care unit at the Uppsala University Hospital to study samples taken from COVID-19 patients. So I'm wondering if you could talk a little bit about this, the sampling process, and specifically the uniqueness of the sampling process of these critically ill COVID patients in this complex post-COVID study.
Yeah, definitely. I mean, COVID, of course, has been a catastrophe for all of us all over the world, but we can most likely learn quite a lot from this pandemic infection, because we know the onset, we know also what is the infection agent, and we also can monitor these patients over time very nicely. So what we have been doing here at Uppsala and some other places, of course, in the world, being focusing on both neurointensive care patients, those patients that have been infected by COVID-19 and or SARS-CoV-2, and then been getting inflammation or inflammatory reactions in the central nervous system, and they end up in the neurointensive care unit then here. And in parallel, we have the other cohort that is not affected central nervous system wise, but they have other organs affected. So it can be a lung, it can be a cardiovascular system, etc. And those end up in the central intensive care unit part of the hospital here. So in total, we have collected approximately 400 patients now, I would say, around 50-60 neurointensive care patients where we have then monitored cerebrospinal fluid, blood samples, urine samples, what we ever could get hold of over time in these patients. And the other 300, and I think it's actually 385 today,
Wow.
of the patients that are in the central intensive care unit, where we have not had reason to draw cerebrospinal fluid, but we have collected blood samples and urine samples, and cells have been isolated and so on for screening. So one could always claim, well, intensive care unit is, well, there's nothing, you don't want to end up in intensive care unit, right?
Right.
It's not a good idea. But when you end up there, you're critically ill, and the treatment is also rather tough. So both the initial cause of your admission to the intensive care unit plus the rather heavy treatment you need to go through, heavy medication, being put in a ventilation unit, for instance, and so on, will have effect on you. So after you have hopefully survived through this process, you have been coping with the situation, you get rid of your infection, and you're starting to heal your damaged organs and so on. Most patients will have some sequelae over time, but due to the natural course of the disease, they will cope with the situation and they will be better. Some will, however, end up with a long-term consequence, and in the intensive care unit group, there would be a combination of the initial infection, cell damage, tissue damage you get, and probably an effect of the treatment that you have to go through to survive. So that is what we call the PUSC group, post-acute COVID group, basically. Then we have a large cohort of patients that have not been admitted to intensive care units. So those are the patients that have been staying at home with their COVID infection, handled the situation more or less well, not been hospitalized during treatment. And those are, of course, a very interesting control and contrast group to the intensive care unit group. But we now see, as you know, large cohort of those patients also reporting with long-term sequelae or long-term consequences. And that's what we call the long COVID patient group that we're also now collecting. So this pandemic, as terrible as it is, it actually has given us an opportunity to monitor what happens post-virally in a large cohort of patients. And we have time-resolved samples, so we can see how things develop over time. We can see what interventions are helpful for the intensive care unit patients. We can also see and hopefully also foresee who in the patient cohort will potentially end up with long-term consequences, and maybe in the end of the day get the diagnosis ME or ME CFS.
It's so important to be able to, we have this opportunity for the first time to kind of see things at the beginning of an infection, which is really important because hopefully we'll be able to get some insight about what happens, maybe, you know, in a genetic function or all these other ways that we have access to study right at the beginning, because I think that is so important with ME/CFS. And oftentimes people with this disease, they are coming to the doctor, they don't necessarily, some of them take years for an actual diagnosis. So by the time they might actually reach a lab or be able to be studied, it's, you know, 5 years post onset. So COVID, I agree, definitely gives us this interesting opportunity that we might not necessarily have had before. So just to piggyback off of that a little bit, so how might this diverse data collected from the COVID patients specifically over these long periods of time, because this is longitudinal, reveal compelling results?
Yeah, so as I said, if we can find patterns or mechanism that could explain why a person end up with a long-term consequence after an initial infection, that mechanism could of course lead us into well, not only diagnostic tools when it comes to specific biomarkers or patterns or sensitivity markers, you could say, genetically. It can also of course lead us into potential treatments and well, hopefully in the end of the day a cure for these post-viral issues that occurs. And I think that another thing that we should keep in mind also, one positive thing with the interest that COVID and post-COVID patient has recently gained from governments, healthcare systems, funding agencies, etc., will hopefully help us to also make the acceptance and interest and understanding of ME CFS patients' situation much better. So that's what we really hope for. As you know, ME CFS patients have been really neglected over all these years, and much more research and understanding needs to be given to these patients.
Absolutely.
And yeah.
Absolutely. And so another question that I had just in terms of the sampling, just because we were talking about it so much, is why is cerebral spinal fluid? I know that's one of your special focuses. Why do we look at CSF? What makes that important when studying ME/CFS patients?
Yeah, so cerebrospinal fluid, as although being an invasive sampling, and I don't know if I should explain how that's done, but it's
that would be interesting. Sure.
Yeah, some patients undergo a lumbar puncture during some of their investigations, so you will know what that is. But what you do is you take a fine needle and you puncture in the lower end of your back into what we call the dorsal sac. And in the central part of the brain, we have a production of a liquid called cerebrospinal fluid, and that liquid is produced actually 500 to 600 milliliters a day.
Oh.
And the total volume in the whole region is 150 milliliters. So that tells you that we actually replenish or change this liquid several times every day. So it's a continuous production and continuous resorption of this liquid. So if we can then draw some liquid, some samples from this liquid, we can actually get a picture, the chemical picture of the central nervous system composition, molecules that are released from the central nervous system into this liquid that surrounds the central nervous system. So a fantastic opportunity to get a chemical picture of the brain without actually drilling a hole into the brain and pulling out a piece of tissue. So that's what I think is so fascinating with this liquid.
It's almost the closest thing to drilling into the brain is the CSF.
Exactly. Yeah. So in some cases, we need to do that also, drill a hole and maybe take a biopsy or put a small catheter into the brain and do dialysis from the brain tissue. So that's done quite in routine now here at Uppsala University Hospital for traumatic brain injuries, for instance.
Oh, wow.
But in other cases, it's the cerebrospinal fluid, the lumbar punctured fluid is very useful for us for diagnosis and monitoring reactions in the brain. So why wouldn't it be enough just to take a blood sample? Well, sometimes it is actually. We can measure things in blood, of course. There's a lot of molecules there. But if we are looking for specific markers that actually relates to the central nervous system, if we try to monitor those in circulation, in blood, we dilute the signal from the central nervous system into a much larger volume. So human adults have approximately 8 liters of blood in circulation, while we then in the central nervous system have, as I said, 150 milliliters. So it's a much more concentrated or brain-specific concentrate of chemical substances in that liquid compared with the blood volume that we have. And if we would monitor something in, let's say, urine or saliva or so, you can also imagine that, of course, easy to get to, non-invasive sampling is possible, but we don't get tissue-specific signals very likely.
Okay.
So.
That makes sense, because I know oftentimes a lumbar puncture is used when trying to diagnose these neurological conditions, but often with ME/CFS, when you go and you present with symptoms such as just chronic fatigue, sometimes people will get a lumbar puncture to see for diagnostic purposes, but not often. It's not often used. Say you just go to a doctor's office, and it's hard to perhaps get that done. So it's good to be able to have a test, I think, that although perhaps more invasive than a blood test, it gives us this more in-depth picture into what's going on in the nervous system.
And that in combination with imaging techniques, for instance, doing an MRI or MRS or PET scan, CT, etc. of the brain, that could of course help us to understand more location and the size of the inflammation and so on. So I think we have to use all the tools we can get hold of, basically, for these complex diseases.
Absolutely, absolutely. And so brings me to the next question, which goes a little bit into what we were just talking about. So if money was no object, and obviously with ME/CFS, it is always underfunded globally. Hopefully that will change with long COVID on the horizon. This is what we hope. But if it was no object, what would you like to do towards finding a cure for ME/CFS and these related diseases?
I think, yeah, still we need to work even more on this, finding mechanisms, finding biomarkers, finding diagnostic, prognostic, therapeutic biomarkers for understanding the process that takes place in the patients. But I think we also now are about to reach the level of understanding so that we should start initiating at least small pilot trials, well-controlled, placebo-controlled trials in well-defined small cohort of patients with different interventions that could be relevant for this patient group. And of course, there's a lot of potential therapies that could be used, but we try to learn from our intensive care unit patients, from our neuroinflammatory reaction patients with other diseases. Yeah, autoimmunity is one other target that we are looking into, and we're trying to learn from other a bit more well-studied diseases to see what kind of interventions would be the most likely to work in our ME/CFS patient group. So I think that is, I would say, the next generation of projects that I would like to conduct here in Uppsala, and of course in collaboration with colleagues around the world. So I think that would be my answer on that question.
Absolutely. I think it's important to borrow from what we already know from other diseases, which is kind of a lot of the work that you're already doing with looking at things such as HSE. So all of this work that you're doing is so important, and we will be very excited to share more as we have updates coming out of Uppsala. And right now it is May Momentum. So if you would like to support the research initiatives and efforts of Dr. Bergquist and colleagues at Uppsala, please consider donating to our May Momentum campaign, which takes place for the entirety of May. And thank you so much for joining us today, Dr. Bergquist.